Dr. Peder Larson is a professor in the Department of Radiology and Biomedical Imaging at UCSF, whose research program is focused on the development of advanced imaging technology at or near translation to human studies. He received his PhD in 2007, after which he joined UCSF to work on hyperpolarized carbon-13 metabolic MRI, and his group has worked on projects ranging from the development of hyperpolarized metabolic carbon-13 MRI methods, leading the translation of hyperpolarized carbon MRI to kidney and heart disease, as well as techniques for imaging connective tissue such as myelin imaging methods, and improving the accuracy of PET MRI and radiation oncology planning, as well as lung MRI. The title of his talk is Open Source Software Tools for Emerging Applications of Hyperpolarized MRI and Lung MRI. And so with that, I'll stop sharing this screen and I will turn it over to Peder. Peder?
Great. Thanks so much for the introduction, Brandon. And yeah, thanks everyone for tuning in. I'm looking forward to speaking on this topic and hearing the other speakers as well. So I'm out in San Francisco here, and I'm going to talk about two areas where either I've been involved or advocated strongly for sort of open science practices. And I would call these are relatively small research communities that are not brain diffusion or some of the fMRI or something. This is like maybe I want to say growing, but hyperpolarized carbon-13 MRI and lung MRI with proton. So in both relatively small communities, but I think it is communities like this that could especially benefit from open science practices and sharing resources, since there are limited number of people and resources. So I'll talk about lung MRI, and distinct from your next speaker, we'll talk about xenon. I'm going to talk about just using plain old protons for the lungs, which has these issues, which is why it's not very common, of low proton density, short T2 star, leads to long scan time, lots of motion. And so I've worked in this area for a bit, and let me walk through each part of how some of the ways we've done lung MRI and where we're sharing. So the sequence side is notably not, well, maybe not notable, but it's not open source. It's a version we use. It's a GE version from Professor Kevin Johnson at Wisconsin. It's kind of widely shared, freely shared by Kevin, but not open source part. So I think an area for improvement. I'll talk a little bit about sequences in hyperpolarized carbon-13. But where there has been a lot of activity in sharing and doing open science has been on things like motion estimation and reconstruction. So this is from my sort of first project in this area where we're looking at, we've got to do things like you have a case-based trajectory on the left. We want to, extract motion waveforms. You want to do some relatively heavy compute reconstructions here. So these are all, pretty non-standard tasks. And so once you've got the data, doing everything after that is had to do a lot of specialized tools, I would say. Just a flavor for one of those is like trying to do this high frame rate, high super undersampled reconstructions to watch different breathing patterns and things like that, image navigators. So so we took this like our first venture into pulmonary imaging and got up on, I just use GitHub. A lot of folks like GitLab as well. I think they're both great. And created this kind of try to create sort of an all-in-one package to take your raw data, do some of these motion estimation techniques, relied on the BART toolbox, Berkeley Advanced Reconstruction Toolbox. Many of you may be familiar with. Has very good features for particularly non-Cartesian data. And the first, lesson that was really good we did here is we posted a data set as well. These are like gigabytes of raw data. There you see 3.2 if you can read real small in there. Very computationally intensive in kind of reconstructions as well. But this data set has been used by I don't know, probably had tens of people reaching out to use this code, and we send them this data set, or they access this to test things out. So that's been huge, and debugging for getting things outside of your own environment. Kind of technique wise, went a little bit further. These are a little nicer looking dynamic image navigators. Always fun to look at, but we're not here to look at those. We're here to talk about open science. So, and this was done by another student, now in Python as opposed to MATLAB. Sample data sets again, couple dependencies showing up here, SigPy CUDA. And then the thing that Frank Ong, the student on this work, did was he used Google Colab. You see right up here, and created a sample way for somebody to use this. And I thought this was great, like super low barrier to entry, right? You can go out, launch Colab very, very quickly and have a nice starter set of code to go with here. And fair warning, I have more slides than time, so I'm going to breeze through a few of these. And then we got into reconstruction algorithms. So again, pretty complex, pretty computationally involved, definitely not, off the shelf stuff. We've added in things like image registration here. So we're bringing in more dependencies of software, but to hopefully do the best job at reconstruction. And more pretty pictures in kids here. Like looking at these, I'm super happy by these results. Okay, but so also again, same ecosystem here. You'll notice in the pretty small up here, but these are all under actually a GitHub organization called Pulmonary MRI that I created. As a thought of where there are going to be multiple tools here, try to make them easy to find, because now a lot of people sharing stuff online, right? How do we find this? And I think there are other good efforts, of course, like through the ISMRM as well for posting toolboxes. But this is kind of going to be a growing problem too. How do you find things? How do you bring in new users to things? Two things I'll point out here. I apologize if it's kind of small, but there's 3 versions of this IMOCO. There's a MATLAB version, there's a Python version, and then that I think did not rely on NumPy, and then one that does rely on NumPy. So this is another challenge sometimes you run into of, well, what language are you going to use, and the pros and cons with that. And this has been a bit of work, I would say, to keep different versions up. And some people have preferred MATLAB, some people prefer the Python versions. Not professional, right? We're kind of hacking things as we go along here, but this is what we've got. But I've highlighted at the right here a couple other repositories that I've forked into this pulmonary MRI organization. And these two were ones that I think they're really built off of the IMOCO. So this was something where I think it was very beneficial that we posted this code, had pretty good working code, had that sample data set still, that original one. And these other MostMoco, ICOMOCO, which just sounds great. These all built off and did comparisons to IMOCO. And so, I think that was our contribution to kind of moving the field forward, is putting things out like this in a way that's usable. And then I've just, for my knowledge, I've tried to create forks of these just so I kind of have, tracking, or people want to compare, they're trying to keep them in the same place. And And we've had other people use this. This is a nice example from groups, folks formerly at Wisconsin-Madison, who were able to get this running on some of their data and get some very nice results as well. And so this package has been, I've been really happy with getting it out to other people and making it usable. The next iteration of the algorithm, we got more motion fields, and I'm bringing this up. There's a lot of image registration going on here. And so, so this is page for that. What I want to highlight here is what I've pulled out is the dependencies. And, this is just one of those things you got to deal with. You got to do it right because and doing it right from the start, understanding what are your libraries, you're using what versions, dependencies, especially with things that are a little more exploratory, like SigPi I've got up here. If you're not familiar, check that one out for MRI reconstruction. ANTS, popular image registration that we used a lot. And again, it's not as big as some of these other ones like NumPy, PyDicom, these really, really big projects. And so keeping kind of good track of these and making that accessible to your other users is really, I think, important in the kind of the lessons learned by this point. So that's my quick journey through our open science and lung MRI. It definitely is extra effort, right, to do this, and to do it first time, to maintain it. But if you believe in your work and you think others are interested in, this is really, it can both, selfishly increase the visibility of your own research, but more altruistically, it can accelerate the field that you're in, by, putting those blocks out there for everybody to build off of. I mentioned the challenge, like I think having good tutorials, sample data sets has been key. Dependencies, versions, man, I hate that, but it's going to be there. And these projects in particular have pretty heavy compute requirements, and so Colab sometimes it's not worked very well for these because it's limited what you get. That's still a little bit of a challenge. I always want to see more engagement from users and community. I don't have good answers for that. And some of these techniques, are at the point where we kind of have them deployed on ongoing studies like here, and that part I think not as good at. I'd love to, maybe there are better ways to work with the vendors or sort of take these out of like proof of concept research tools and do that kind of in an open fashion as well. I would like to see more, and I know there are pathways to do that through some of the vendors as well. Yeah, I'm just going to keep on going into hyperpolarized carbon-13. Please drop any questions you've got in the chat, and hopefully we can have a few minutes at the end. So the background, quick, your crash course in this technology is these hyperpolarized, so we do carbon-13 enrichment given the low natural abundance of carbon-13, and we do this hyperpolarization process to increase the signal, basically, and sort of fortunately, there's this agent pyruvate labeled with carbon-13 that sits at this very interesting metabolic switch where particularly cancers will, as on the right, will preferentially create lactate from pyruvate as opposed to going into the TCA cycle, this Warburg effect. So this is what the experiment looks like very quickly, circa 2010. Polarization for about an hour, rapid dissolution for 10 seconds. Younger version of myself takes the contrast agent into the scanner. Somebody who's more qualified to work with animals injects the agent into an animal at that time. And we do rapid imaging here. Okay, and then this is where we need to be aware of a little bit what's going on. There's a time scale up here. So this is all happening quite rapidly. It's on the order of seconds. We're up here at, coming up on 30 seconds, and signal is getting pretty low. We're looking at different metabolites, pyruvate, alanine, lactate in this case. So we've got different resonance frequencies that we're trying to image almost simultaneously or in an interleave fashion. So these are some of the challenges that we're going to have to deal with in this technology, and this is where these specialized tools come in. Come back to that. And then 2014, okay, images look a little better. Doing a lot of I think 13, 14 sites now doing human studies. So it's grown a bit. It's still, relatively small community, but it's grown a decent amount. So and again, I'm going to blaze through a few of these, but this is the whole pipeline. I'm going to be focused more on the right as to where we can do open source work, and it's most of it. I've tried to advocate for the centralized toolbox that has a bunch of functions in it that I'll go through. So done a little bit on the calibration bit here. So we use this RT-Hawk software from Vista AI, formerly Heart Vista, to do some of like automatic tracking and calibration that are going to be shown in the movie here. And this has we have this sequence code available. The problem is not everybody has this system. It's sort of the hardware and software required is kind of a high barrier. And so doing making this an open tool has not panned out as much as I would have liked. But I'll talk about sequences and the acquisition a little bit here. So different resonance frequencies, fast decay, chemical shift, well, lots of stuff to worry about. And this is our preferred method, this frequency selective echoplanar imaging with spectral spatial excitation. Now, on the sequence side, just the past year, many of you have probably heard of PulseSeq now, sort of an open source framework for doing vendor neutral pulse programming. Support at least on GE and Siemens when I made this slide. A very, very nice tool, and I think getting a lot of traction in the community. And so we tackled it could we do this for hyperpolarized carbon-13? The thing we had to do is the multinuclear spectroscopy, MNS, so non-proton imaging was not supported in the GE interpreter. That is now, there's a version of that now supported in the GE interpreter called TOPI. So that's available to anyone doing multinuclear stuff, and a few sequences that are actually pretty easy to make in this this software. It's really great, and there's also another package, a group at WashU called XEPI, a really nice design of some of these types of sequences. And it's, these are great because you can pass them around on different scanners and different versions or types of scanner. And so I like this approach a lot, at least, or one of the best options I think we have if we want to do cross vendor and keep things consistent. What's in that toolbox is something I did a long time ago of spectral spatial pulse design. Has been one of the pretty popular features in there. And this is pretty old, but it's been up there for a while. It's still being used, so I'm pretty happy about that. It has a lot of demonstration scripts, and over the years I've tried to make these simpler and simpler because they're like overly complex and just for what needs to be done, there actually can be quite simple. So that's the sequence bits, and then the other side of the hyperpolarized carbon that is unique is how do you deal with this data? It's multiple spatial dimensions, metabolite dimension, temporal dimension. You're not going to just natively get a single image out. That there's really going to be interpretable or reproducible. So there's a number of different things you can do, a number of things people do, kind of varies. I've kind of advocated for this pharmacokinetic or pharmacokinetic model because they have some nice biological basis you can build into them. You can build in some of the MR physics, T1, flip angle effects. But sometimes these, in different people's hands, like these models can be a little finicky. You got a bunch of, you have free parameters or you have assumed parameters. And so a few years ago, set out to create some methods that would just be like robust and at least the baseline implementation would be robust and pretty simple to run. So kind of looked at that from the theoretical point of view and then put this in this toolbox. And I think because of the reason that it was just, I mean, maybe it was out there, but also like the first version you touch, if you don't do anything special, is going to work very consistently and very stable. And so this has become, I think relatively well used within the hyperpolarized carbon community for some of those reasons. Couple of the features that I think have really helped here as well is distribute sample data, and these are just in line with the toolbox because they're much smaller than our lung data sets. We have sample code and Jupyter notebooks running Octave to process the sample data, and so you're just trying to keep that have some benchmark people can run, have something simple people can come in and do from very quickly. I also use this maybe my binder service, which is well integrated with GitHub, at least, and it will launch Jupyter notebooks and run on their sort of donated compute that this service has. So that's also creates a very low kind of barrier for people to get started and use this. And some other fun stuff, building in some metabolic phantoms is kind of a nice thing you can have for benchmarking, testing things. And so here's my lessons learned from the hyperpolarized carbon-13 work. In this field, in general, requires a huge amount of patience. It's a very slow-moving, very challenging experiments field. And even with the toolbox and these things here, for many years, I really had to just put them out there on the hope that somebody would find them useful at some point and just believe in that and be the champion and keep committing, keep updating things. Any chance I heard of feedback, I integrate that and just be pretty persistent, honestly, of saying I think this is a good way to do things, and I think now it's that has succeeded, and it's succeeded particularly for the parts that are very specific to this technology, things that people can't find elsewhere in other tools, or maybe there's not more general purpose tools for these things like the spectral spatials and the kinetic modeling. At this point, it's the problem is that it's old and the code base is kind of messy, and it's all MATLAB. Should it be Python maybe? So infinite time and resources would do a lot of these things, but that's my wish list there. And just a couple of general takeaways. I mean, GitHub or GitLab, seen it done well both ways. Get your DOIs from Zenodo, both for your code and for data sets, so you can cite these things. So you get credit, and both so you can track usage, so you can easily hand them around. And if you use GitHub, GitLab, there's amazing features in here: issues, pull requests, access control. Might you get literal recognition? I mean, this can make your life easier by doing GitLab, and of course using version controls is a must. And then I found a lot of mileage from using the notebooks, so Colab, Binder. And if you haven't seen NeuroLibre, which publishes papers that are notebooks, I would definitely check that out as well. And that's it for me. Thank you all for your attention.